Hi, and welcome to the AP Biology Lab 3 Mitosis and Meiosis podcast. Both parts of this actually require a microscope to do, and so I'll get that out of the way. We actually don't do the second part of this normally, and that's just because we haven't had much luck using Sordaria, but I want to make sure that you're ready for the AP test if they ask you questions about that. Mitosis and meiosis. First of all, we should talk about differences between the two. Mitosis, if we take cells like this and we want to make identical copies of those, we can just simply copy the two homologous chromosomes, in other words, the one you get from your mom and the one you get from your dad. We can make copies of them. They'll look like that. They'll eventually line up along the metaphase plate. We'll pull them to either side, and eventually you have two somatic cells that are exact copies of that original parent cell. That's mitosis. And that's how you go from that first zygote you were when you were just fertilized egg to the billions of cells that you have inside your body today. Each one is identical. DNA is exactly the same. Now, meiosis is a little different. Meiosis, what we do is we actually start the same way. We duplicate those chromosomes. But here it gets crazy. In other words, the homologous chromosomes, again, come together. There'll be crossing over that occurs, and eventually you get 4 cells instead of 2. Now, how do those 4 cells differ from the 2 cells that are made in mitosis? Each of them are different. And so each of them, due to crossing over and due to that independent assortment, look way different than in that original parent. And that's the whole function of sex. The function of making gametes in sex is to make sure that next generation is different than the generation before that. Okay, so let's start with the mitosis side. And we do this in class. We could either look at onion roots in this case, or we could look at a whitefish blastula, but we're going to find similar ratios. In other words, we're going to find cells right at the bottom of a root that are actually dividing through mitosis. They're going through interphase, prophase, metaphase, anaphase, telophase, cytokinesis, and then that eventually, as they get larger, that's going to push the root down through the ground. And so you can actually see this is a root cap on the top that actually protects it, but behind that we can actually see cells that are already in different phases. Or we could look at a blastula, which is essentially a baby whitefish, stem cells as they start to divide, which eventually is going to create that new fish. Okay, so we can look at either place, but essentially when we look at either place, we're just going to have one person checking boxes and the other person looking underneath cells at the microscope. And so one person will be filling in a data chart like this, and the other one is simply going to be looking at underneath the microscope at all these different cells and going to be counting it out like this. Interphase, 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 prophase, interphase, 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 prophase, interphase, 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 anaphase, interphase, 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 anaphase, interphase, 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 prophase, prophase, metaphase, interphase, and the other person is just feverishly writing those down, checking boxes as quickly as they can. And so you can see in this year we counted, how many is it? 2,000 total cells. And if you do that, you can figure out the percentage of time they spend in each of those. And so we then graph that on a pie chart, and it looks like this. And you can see that 73.8% of the time it's actually a cell in interphase. Now, that's important. In other words, they spend much more of their time in interphase. That's because a cell has to grow, it has to duplicate its DNA, and then it has to grow again. But we could take those percentages and actually add them to time. And so that's this fourth column right here. We're multiplying those times the number of time in each day, and we can figure out the number of minutes they spend in that. That's the mitosis side. The meiosis side we don't normally do, but you should understand what's going on. You should understand this diagram. In other words, this is mitosis, and in meiosis, and in meiosis you're making gametes. In this case, those are spores for a fungus. But we can either do crossing over or we can do no crossing over. And if we do crossing over, you're going to get a different spore arrangement than you are if we don't cross over. In other words, if crossing over occurs, those homologous chromosomes actually swap bits of them, you're going to get either a 2 to 2 to 2 to 2, or a 2 to 2 to 2 to 2, or a 2 to 4 to 2, or a 2 to 4 to 2 arrangement. That means that when these two fungi, and I should have said that a second ago, you have a tan fungus and a brown dark fungus, and as they grow next to each other, they produce these spores, which are a hybrid of the two. You then squash them out, and you can see these different spores. But in those creation of spores, or in meiosis, if you get a 4 to 4, that means that no crossing over occurred. And so we can look at this and we can do some stats on it. In other words, this is a 4 to 4, and so this one, there's no crossing over. Or in this one, it's a 4 to 4, so there's no crossing over there. But on this one, you can see it's clearly a 2 to 2 to 2 to 2, or this one's a 2 to 4 to 2. And so we could say that those two actually show crossover. So I could count the whole thing on here. I could figure out how many have crossed over, how many haven't, so I could figure out what percentage of those are crossing over. And remember that frequency of crossover is important because it tells us how far those genes are on that same chromosome. And so that's the meiosis lab. Again, it's one that we don't deal with a lot, but you should be able to look at a slide like this and count the ones that are showing crossover. And you should be able to understand this diagram over here and really understand what I'm actually talking about. So that's mitosis and meiosis. I hope that's